All right, guys, lab number 7, seedless vascular plants. We already just talked about those, some of the club mosses. And today you will see like a podium and selaginella, and this is the club mosses, and they are important because one of them will be something which known as a homosporous. See, same kind, the spores are the same size. You're looking at the sporangium, the spores are about the same. Versus heterosporous, when you're looking at the sporangium and you will see big spores and the clusters of the small spores, megaspores, microspores. So make sure you will be able to identify that, and I'll again pointed it out for you as well. So let's kind of take a look. We discussed the psilotum, the whisk fern, which is resembling. It's a vascular plant, but it's resembling something which known as the one of the first plants we know of, known as the Cooksonia. So we have the dichotomous branching out stems and the sporangia. And then, of course, the sporangial cluster of sporangium known as the strobulus on top of the horsetails. So let's take a look. Question number 1 on the pre-lab, right? 1, do seedless plants exhibit alternation of generations? Any plants do. So yes. Is the dominant plant body for seedless vascular? As soon as you start saying vascular, and if you see it, vision, vascular vision, it's a diploid. If you can see it with your naked eye without the microscope, it's most likely diploid. So it would be a sporophyte. Sporophytes are diploid. Leads to critical innovation that leads to the success of the seedless plants, and you can find it somewhere around here, right? So we are talking about there should be vascular tissue and the windblown spores. This is where you can find that stuff, but vascular tissue is the predominant thing. Do the lycophytes and pteridophytes reproduce by spores or seeds? It's called seedless vascular plants. They called seedless because they're not producing seeds just yet. Therefore, it would be spores. And then, of course, spores are haploid. Name 2 major phyla of seedless vascular plants, and we are talking about the Equisetum and the Pteridiophyta. Pteridiophyta and Equisetophyta. Lycophyta also club mosses. So any of those will do. Is the strobulus, a spore-bearing structure of the club moss, found on a sporophyte or gametophyte? That's a good question. Think about it. It's a spore-bearing structures. It will be found on a plant which bears spores. And here we go. It's a sporophyte. Now, what is the difference between the megaphylls and microphylls? Well, size, mega and micro. Mega means large, micro means small. Now, what's a phyllis? Leaves, tiny little bitty leaves or large leaves, like fronds, megaphylls. And then the kind of tiny little bitty leaves in the selaginella. You will see them in the dried up, well, in the herbarium specimens. Are the gametangia, the structures which producing gametes, Antheridia and Archegonia, found on a gametophyte or sporophyte? So gametes would be found on the plant which produces gametes. Phytos means plant, and the gametes, gametic plant essentially. So it will be found on the gametophyte. So now let's take a look at the table 1. What you will see is actually several slides, the microscope, which will show you that strobilus, it's a T top. This is where the spores are located. Strobilus are clusters of spores, sporangias, under the microscope, so you can see a high magnification, and also a dissecting scope, so you can see the whole thing. And then, of course, the you will see the dried up specimens, the herbarium for it. So we are looking at the specimen right here for the club moss. And again, this is the underground stem, rhizome, with the stems and the tiny little bitty leaves, microphyls coming out. And then on the top, you will see those particular club-looking structures, and those club-looking structures are strobili. So the plant body of the lycopodium, that's a lycopodium right here, haploid or diploid. You see it with your naked eye, therefore it will be a sporophyte. It's a diploid, which is the next question. If it's a diploid, it's a sporophyte generation. Does this generation bears gametes or spore? It's a sporophyte. It bears spores. That's why it has a strobili on top. This is where the spores are produced. And this is the next question. Where the spores are produced? In the strobili. So look at the slide of the lycopodium strobilus right here. And you will need to label, yeah, for that one, you will need to label rhizome, stem, microphyls, and strobilus. In this case, you will need to, I think it's moved. You're supposed to have that thing over here, probably. Right there. And then it should go like this. Sorry, guys. Yeah. So here we go. So the whole thing is the strobilus. Sporophylls, those tiny little bitty leaf-like structures which are surrounding the sporangium, that sac with the purple stuff inside, that's the sporangium, a single one. There is a bunch of them. Okay, so the whole thing is the strobilus. Sporophylls, again, that's a tiny little bitty leaves which is surrounding the sporangia. And increase the magnification. So here we're looking at at the increased magnification, look at those. You can see the sporophylls, right? And this is the sporangia. Now look at the spores. The spores are all about the same size. You're looking from one sporangia to another sporangia to a third sporangia. All the spores are about the same size. So homosporous or heterosporous? Same size. Is it a homosporous or heterosporous? Everybody see the about same size spores, right? So type it in. Tell me, homo, hetero, homo. Yes, that one is the homosporous. You're looking at the picture. A single dot right here is about the same size as the single dot right there, but also in a different sporangia, it's about the same size. Maybe not exactly the same size, but pretty close. In a different one, you will see that in one sporangia, you'll have a huge spores. And in another sporangium, you will have a tiny spores in the same strobilus. And that would be a heterosporous. In a Selaginella, you will see it momentarily. But for the lycopodium, the spores are in a different sporangia. The spores are about the same size. Therefore, that's a homosporous. Thank you for the correct answers. Oops. So here we're talking about the sporophylls, leaves. We just talked about that, and the spores. And you can see in the different sporangia, the spores are about the same. So next one showing the Selaginella, a much smaller, and that one is a really look like a moss. And you will need to take that picture and, or better yet, this one. And then you will need to label leaf, stems, roots, and strobili. So stem, all of that is essentially a stem right here. Those ones, the thinner parts, that would be roots, of course. Leaves are almost impossible to see because they are tiny little bitty ones. And then on top, you will have a strobila right here. Now, when we start looking inside of the strobilus, look at this. We have a strobilus overall, and we have a different sporangia. Here we have a sporangia which contain only 2 huge spores. That sporangia contains 2 huge spores, but that sporangia in the same strobilus contain tons of tiny little bitty spores. On that particular picture, you will see the sporangia, tons of small spores, sporangia, tons of small spores, sporangia 1, 2, 3 spores, but huge. Those ones are known as the megaspores. Those ones are known as the microspores. And therefore, that would be a heterosporous structure or heterosporous plants. Make sense? Any questions, any problems? Same size spores throughout the strobilus or different size spores throughout the strobilus? Different would be hetero, all the same size as we said before, that would be a homosporous. Is Legionella homosporous or heterosporous? Well, apparently it's a heterosporous. Are the spores haploid or diploid? That's a great question. So tell me those spores, specifically megaspores, haploid, diploid, type it in your chat box thingy. Homo, hetero, depends. So haploid, diploid, spores. Okay, Maria answered something. Anybody else? When we started lab number 5, actually when we start talking about the lecture before the lab number 5, when we start talking about the fungi, I told you guys, please remember, spores are haploid. Fungal spores are haploid, plant spores are haploid. Megaspores, microspores, homospores, heterospores don't matter. Spore, myospores, zygospores, ascospores, basidiospores, as long as it has a spore, spores are haploid. Megaspores are haploid. Microspores are haploid. All right? Please, haploid. I don't know how many times I told you spores are haploid, and then I'm saying, hey guys, what about the megaspores? And then you start doubting yourself. Don't do that. You know this stuff. Megaspores? Okay, spore haploid, moving on. Which trait is more advanced, homosporosity and heterosporosity? Well, I will tell you heterosporosity is more advanced. And we need to explain the answer. Well, because I said so. No, that's not a good explanation. Think about it. Megaspores will grow, it's larger, it has more nutrients essentially. It will grow into a female organism, the one which will produce eggs. So the nature literally taking care of the females. They are more important in the reproductive cycle. I'm not trying to put down guys whatsoever. No, it's just from the reproductive point of view. The females, the guys are donors. Males are donors. They donate and they forget about everything else. The females are the one who's stuck with the plant kids, with the embryos. They have to produce the gametophytes. They have to produce, well, not gametophytes. They have to produce sporophytes and so on and so forth. So in this particular case, taking care of the females in plant kingdom, and later on we'll talk about the animal kingdom, is a very crucial step from the point of evolution. All right, make sense? Moving on. Table 3, whisk ferns. So here we have microsporangium, microspores, megasporangium, megaspores, and now we're looking at the fern, the actual fern plant, and then there will be here we're looking at the psilotum. That's the actual something which known as the whisk ferns. They are growing not a very tall, and again looks very similar to what we had at the dawn of the plant kingdom. That's a Cooksonia. Of course, we don't know how it looked like, but we are thinking based on whatever we recovered from the fossil record. This is how Cooksonia look like. This is how the psilotum look like. So observe and sketch specimen. So you can take a print screen that, and then bearing the spore bearing structures, those particular yellow dots, this is the sporangia. Do you see sporophylls, leaves surrounding the sporangia? No, they don't have leaves, they just stem with the sporangia. Is the plant body of psilotum haploid or diploid? You can see it, it's about 10 inches or so. You can see it with your naked eye. You start seeing naked eye, vascular plants, that's the diploid organism, also known as the sporophyte. Diploids are always a sporophyte. Haploid are always gametophytes. All right, moving on. Any questions, any problems? No questions, no problem. All right, so table number 4, horse tails. Whenever you're talking about the horse tails, this is, I guess it looks like a horse tail. So in this particular case, it's kind of horizontal laying down. So you will see the stem with the whorl of leaves coming out. And on the very top of that, you will see the sporangium, or cluster of sporangium known as a strobilus. So strobilus on the very top, higher from the ground, so the wind can easily pick up those particular spores, the stem, the central part. So here we're looking at a stem right here. That's a good picture. So here we go, dried up horsetail. This is the stem. The part which is coming out are leaves, and on the very top right here, on the very top right there, you will see the cluster of sporangia known as a strobili. It's basically a pine cone in the making. Not a pine cone yet, but kind of in the making. How does leaves are arranged in the equisetum? Here we have that's the word, whorl. More than 2 leaves coming out from the same node. Is the plant body haploid or diploid? Again, those particular things can grow up to 5 feet, 4 to 5 feet tall. Clearly can be seen with the naked eye. They are vascular, therefore it's the diploid organism. As soon as we start talking about the diploid, we're talking about the sporophyte. Where are the spores located? On the very top, the structure known as the strobilus. Easy enough. So here we're looking again. Look, this is the stem, leaves, and then tiny little bitty strobilus on top. Here we have a stem, tiny little bitty leaves, and fairly large strobila. So again, sometimes they don't have these leaves. The leaves, tiny little bitty microphyls, remember, small one, and then the strobilus. So now we're looking at the ferns, different species of the ferns, of course, the green ones. So you need to sketch. I'll take a picture. Fiddlehead, you cannot see the fiddlehead right here, but you can clearly see the fiddlehead and it literally looks like the violin, head of the violin also had the same violin, fiddle, pretty much the same thing, right? And the whole thing is the front. On that particular, on the front history, you can actually see the tiny little bitty sporophyte growing, and you can see the preserved fiddle heads. They are young shoots, or technically young fronds, young leaves, and they are quite nice. I mean, you can eat them, no problem. I haven't tried the pickled ones, but apparently you can buy those ones. So now sporophyte, we already, any of those pictures, that's a sporophyte, that's a sporophyte. The gametophyte, you will see it under the microscope. So if it's a sporophyte, it would be a diploid generation. So, yeah. So it's a diploid generation, and because it's a sporophyte, it will be producing spores, right? Now, the spores will be produced in those particular dots. That's the clusters of sporangia, and that's known as the sorus in the singular, two or more, plural, known as the sori. And what they will see, you will see the front, and if you'll pick it up the front on the back side of the front, on the bottom part of the front, you will see the sori. Spores are produced by sori. So we have a sori spores. Spores are haploid or diploid. Type it in, tell me what the spores are. Haploid, diploid, haploid or diploid. The one which produced by the ferns now, the sori spores, so myospores. Yes, spores are haploid. General rule of thumb. Okay, good. So this is the cross section. So we're looking at the leaf, and it's cross-sected through the sorus. Right here, you can see that this is the top part of the leaf, this is the bottom part of the leaf, and that's the sorus. Now you can see that it's a cluster of sporangium coming out, and that's a single sporangium with the spores right there. So from the whole thing is, well, the top part is the leaf, and the sorus would be that particular structure, a cluster. A single circular thing would be a sporangia, and the spores are the stuff inside. Here we're looking at the, we'll talk about the fossilized ferns. I don't think I have it in my collection here, but the fossilized ferns in the cross section, you can actually clearly see the vascular tissues, and you can understand that it's about yay big, so it was a tree fern at some point of time. So now you're looking at the different fronds, different species, and again, you're flipping over the leaf, and you can see those sori. So here we're looking at under the dissecting scope, and more under the dissecting scope, and you can start seeing the single sporangia. Again, single sporangia, all of those under the dissecting scope, you can see the sporangia right there. And now it's a regular microscope, and you can see the single sporangia in the sorus. Here we go. That's the cross section, similar to what we see right there. That's the sorus and the sporangia, and the spores are inside. Here we go. That's a single sporangia, and you can kind of see the spores inside. Now we're looking at the Irish Valentine. Now we're looking at the gametophyte. Gametophyte looks like a heart, I guess, shape of the heart. And on the middle part, you will see the archegonia, larger structures, because they contain a large cell known as the egg. And then on the side, typically, you will see the antheridia. The structure produced tons of sperm. The idea is that you have another gametophyte right next to it. The drop of water will be able to transfer the sperm from one to another to do the cross fertilization. So let's kind of take a look. So the whole body known as the prothallus, right? Then we have a root-like structures known as the rhizoids right here. So the whole thing is prothallus. Irish Valentine. You can use any photo for labeling. You can use, well, technically for the saurosporangian spores, you can, that particular photo is already there. So yeah, all you need to do is say, well, kind of, where is it? Insert. And you guys, lately you are doing a wonderful job on labeling. I really like what you're doing. Different people does different things, but now you can, you're inserting, some of you inserting the shapes are really nice, like very thick ones, and I can clearly see it where it's labeling, like those ones, right? Kind of, you can say one. And then something like this. I see students doing something like that. Yeah, sporangium and then myospores, usually a smaller one. And yeah. So thank you very much. Much clearer, much easier for me to grade. And some of you still using the spider web of the arrows and they pointing everywhere. And sometimes it's kind of hard to figure it out where they're pointing at. And myospores, of course, any spore right here in the sporangium, they will be produced by the meiosis, hence the name myospores. And therefore, well, the spores are haploid. Now, going back to the prothallus, Irish Valentine. So here we're looking at the body of the gametophyte, haploid structures. The central part will have the archegonia with the eggs inside, and the outside antheridia, so those ones will contain the egg. Different gametophyte called, either you can call it prothallus, or you can literally call it Irish Valentine. I will accept. Okay. Is the structure free living independent of the sporophyte? Yes, it's the photosynthetic, it's free living, but it's tiny. You have to have a microscope to see it. It's a 40 time, if not 100 time increase in magnification for this one. So it's very small, literally a speck of green somewhere on the floor or on the forest floor. What is the function of rhizoid? Rhizoid will hold this particular structure in the soil and absorb some of the water. What type of reproductive cells does archegonium produce? Very easy, that one, right? Oh, you're supposed to have egg. Yes, I understand that, EGG, but still kind of gives you the nice visual, what kind of cells it's producing. Eggs. What cell antheridium produce? Well, that would be sperm because there is no eggs inside, therefore it have to be a sperm. Where the fertilization occur, antheridium or archegonium? Remember, archegonium will contain the egg. The antheridium will produce sperm, which will have to swim through the water to reach the egg. So where the fertilization occurs? Fertilization occurs where the egg located at, and egg located in the archegonium. Make sense? All right, next table. So yeah, you can actually see it at a higher magnification, antheridium. Well, antheridium right here, all of those dark dots. And that part is archegonium, much larger structure because containing larger cell known as the egg. Now, table 6, talking about some of the fossils. So in this particular case, calamites are very similar to what we had to modern horsetails. And I'll show you some of the examples of why exactly we think that it's a horsetail. That particular thing right there is a fairly nice piece of the calamite, about 15 or so, 20 inches long, about yay in diameter, about 3 or so inches in diameter, and that's basically a fossilized stem of what? Of calamites. Calamites are relatives of modern horsetails. So first of all, does this fossil comes from a vascular or non-vascular plants? Well, think about it. What kind of lab we have? Vascular plants. So it is a vascular plant. Have similar, how the fossil similar to the specimen in the room. And I'll get you some of the pictures. So here we're looking at the picture. See, that's the part of the stem with the ridges. Ridges, we talk about the hollow stem at the horsetails, hollow stems with the ridges to make it stronger. And now if you look at this particular fossil, which is quite nicely, here's the stem coming out, tiny little bitty stem, right? And then the leaves coming out from the node in a whorl formation. Here again, you're looking at the stem coming out and bunch of leaves coming out. Very similar to what we see for the horsetails. So how is this? And same kind of idea, stem, several leaves coming out, stem, several leaves coming out, stem, several leaves coming out. So in this particular case, those ones are most likely the ancestors of modern horsetails. So yeah, same kind of idea. Look, stem, and then we have several leaves coming out. And then you clearly see the ridges. This is the cross section from the fern, and you can see the vascular bundles. So it's clearly a vascular stem. And here we're looking at the calamites. This is how it's horizontal. Now it's vertical. That's the stem. Again, ridges. And look, this is the nodes. This is where the leaves were coming out. It's pretty much a piece of coal nowadays. As a matter of fact, our department head, a former department head who bought that particular fossil, she bought it from the coal miner. He literally found it in the coal mine, and instead of just dumping it and burning it, he preserved it and sold it, I think, on eBay. So yeah, that's clearly, you can see that it's a fern, oh, it's a horsetail, and the horsetail, very large horsetails. That estimated it was about 15 to 20 feet tall horsetails. And now we have tops of them are 5 feet tall, usually within like 2 feet or so. All right. And the post-lab questions. Let's take a look on the post-lab. You observe two types of the Lycophytus, Lycopodium and Selaginella. Which one is the heterosporous? Selaginella have a sporangia with the different sizes of spores. The large one, the megaspores, and small ones, microspores. Which one is a homosporous? Lycopodium. Spores in the different sporangia were about the same size. Which character is more advanced? We decide that taking care of the females is a good thing to do in life. So heterosporosity. Is the dominant plant body of lycophytes, pteridophytes, sporophyte, or gametophyte? You start seeing vascular plants, any vascular plants, you see it, it's a sporophyte. Do seedless vascular plants exhibit alternation of generations? Any plants exhibit alternation of generations. Any, no matter. Seedless, seeded, vascular, non-vascular, don't care. All of them exhibit in one or another way alternation of generations. Sporophyte producing gametophyte, gametophyte producing sporophyte, and so on and so forth. Alternation of generations. What type of reproductive cells produced by a strobili? In the strobili, we have sporangia, so spores. Spores, again, we were talking about spores being haploid, so it will give us haploid generation, and haploid generation also known as a gametophyte generation. A fiddlehead. A fiddlehead, you clearly can see it with your naked eye. Vascular plants, it's a sporophyte, right? Do meiospores, so spores, germinate to form sporophytes? Sporophyte diploid and spores are haploid, so no. The spores will produce the gametophyte generation. What two reproductive structures would be found on the fern protalus? Antheridia producing sperms and the Archegonia producing eggs. Is the protalus haploid or diploid? Protalus is the gametophyte. It's producing gametes through the mitosis. It have to be haploid. Leads to critical innovation found today in the plants. We talk about the vascular tissue and wind-blowing spores. Name all the plants you studied in the lab today and identify one distinguished feature. So we're starting with the lycophyte, right? Lycopodium. Lycopodium, homosporous plants. Then we talk about Selaginella, which will be a heterosporous. Then we talk about whisk fern, Psilotum, resembles Cooksonia. Then we talk about the horsetails and whorled leaves, and the hollow stems, nodes with the leaves coming out. And finally, we talk about the ferns with the fronds and the sori on the back of the fronds. Oops. Well, reflection questions. I start deducting points for not following the instructions. I need something. One sentence is not a paragraph. I literally still have students saying, what the main purpose did you do in lab? We did ferns. We studied ferns. We observed ferns. We observed seedless vascular plants. In lab 7, we observed seedless vascular plants. Not good enough. Make sure that you will briefly describe what we did in this lab. We talk about homosporous, heterosporous. We talk about different ways how the spores are sending the different spore structures. We talk about the strobilus versus sorus or sori. We talk about the vasculature system. What learn and observe? Same thing. I observed seedless vascular plants. Not good enough. How can I use? I don't know how can I use. Not good enough. It's an open-end questions. Many students hate open-end questions because they don't know if they're writing it correctly. What's the correct answer? Well, you tell me. It's your understanding of that particular lab. Can you apply it later on in real life? I already told you about the call. Clearly can be used in the real life. Global warming, you can tie it up with the global warming if you think about the global warming. If you don't believe in the global warming, there is a climate change and we're contributing to it. Because of that particular plants, which died long time ago, we're digging out, we're burning them. You don't like any of the ecology stuff. We're using electricity, right? About what, 40% of the electricity in the state of Texas comes from fossil fuels, including coal. So because we are right now talking with the help of the computers which are using electricity, we're using those guys. You don't like any of that thing. You can say, well, I really would like to try ferns. Well, I already tried it. My favorite dish in the ferns is blah and blah. I really like the sauteed ferns with the meat salad. It's a traditional Korean salad. Really like it. Tastes good. But again, it's up to you how you can apply this concept or use or modify. You can say the lab was horrible because we didn't cover blah, blah, and blah. I would like to see more of blah. You don't have to only apply or use it in the real life. You can say, it would be nice if we can do this in the lab. I'm reading all that stuff. So since I'm reading, please write something. And once again, that's not just a handful of students who are not really following the directions. Most of you are doing an awesome job. Thank you very much. And I really like that you guys are using your answers, you're typing your answers in a different color. Some of you using purple, some of you using blue, green, red, whatever it is, it separates your answers from the questions. And it's really helpful. It's easier for me to read it right away. Visual representation is important because we are visual creatures, right? All right, on that particular positive note, I will stop today.